I'll set a timer and just try to keep time and our each looking at you and that may be
Thanks, Hao. Yeah, I'm Jun Li here. I'm giving this talk on behalf of all these co-authors to just give an update report about for more than a year, we continue to characterize the revamped genome. I talked to Junior Arthur on that side, and then Hao and I are responsible for the eventual wrapping up of the manuscript, but you can see probably half of the authors are attending this meeting. I don't have a slide on their affiliation. I'm too busy to read. Just want to acknowledge your contribution. I remember when we were meeting in Glasgow, some of us had to get out of the meeting to write that grant to beg Jonathan and Amy for funding. Afterward, many others contributed. So this manuscript, the version from preprint have been posted late September and then submitted to the manuscript out. So it's a good work on many parties, including not only Rob and a folks from RGE. Okay. So I'll refer to BN7. That's the newer genome that I'm aware of. Several efforts to have the next version. They call it a newish genome. In Glasgow, somebody coined the term IROC. Also, it's taunting the mouse people without IMOC. Yeah. So that's one reason I liked it. Then we joined forces with BGP with folks in UK who work in the Shell-like project to help sequencing and finishing the new rat genome using a combination of design technologies. Some of them are should better be called scaffolding technologies. The product has been well, many of you have seen this. I'll just go quickly. Between 6 and 7, there is a dramatic decrease of the number of contigs and increase of the contig N50 and L50. So by several metrics, the continuity of this product is now on par with by then latest human genome. Of course, now human genome marched on to 2 or 3 additional concerns. One is T2T, another is human pan genome. So we are still not there. So my presentation is about the structural description of the rat genome. In many ways, it's an outlier in this meeting is the quantum work in the basement about how the structure may have shifted and how all of us can migrate to the new reference. There was one description I put this out from the manuscript. It's a 6 to 7 line graph documenting segmental correspondence. Most of the green are well behaved, almost like preserved symphony. I can't use that word. The blue lines document intra or interchromosomal shifts. I don't call this inversions or structural variation because these are 2 renditions of the same reality. They're just discordant reports of what what brown genome is like. There was one example you are all familiar. That's in chromosome 6. There is this reverse orientation, again, avoiding the word inversion. So version 6 neglected to notice there was a chunk of assembly that's wholly on the wrong orientation. So this is discrepant between 6 and 7. But then we bring in other data, including what I don't show, but in manuscript, how there is a version of genetic mapping data based on pedigree-based counting of segregation. That new genetic map is consistent with the version 7, but not 6. So the arbiter for a lot of these regions, we can also see the smaller reverse orientation. These are all, by and large, we say 7 is a winner, 6 is in the wrong. But then many of you care about the question on when to move, right? Living in a house, you move to a new house is not trivial. some field left over as the utility engine can do a wholesale conversion of coordinates from one to the next. However, in the back of this utility are these dot plots, genome, assembly to assembly correspondence. By the look of it, they are okay. Probably more than 95% of genome should be translatable or relatable. But in reality, when we give it a try, we sampled one variant per kb, so sending 3 million to leftover, asking how many you can give us the coordinate in the 7. We have 8% loss. So this is because behind this utility or the community service engine, the behind the curtain lookup table declined to deal with one-to-many and many-to-one mapping, so only offer a solution of one-to-one. So if your work or if your favorite finding suffer misfortune or fall into a percent, I think the best hope is to do alignment with the dot plots yourself to know the balance of the situation. So that's kind of advice. It's not super helpful, but it's a practical tip. This number 8% is also not generalizable. If your data set are gene-centric or recombination-centric, you'll get a big number. Then the manuscript as a descriptive thing about improvement of the new transcript, we offer many vignettes. One of them is to say what used to be declared as a transient KPL in the R6 by the new mapping, by the new coordinate, turns out to be proximal to the gene, therefore reclassified as a cis KPL. Then we move on to the data in hand. There was a whole genome sequencing short read data on 163 animals. They now give you the variant type, but I keep thinking about which user will care about which part of this, because some care about string variation, others about the two-parental matching population, others care about the super close substrings. So depending on your scope of your view, you care about different variant sets, and then their impact on genes may also vary. There was a global view. This is the colored IDS matrix. The green blocks are high similarity. So from there are green blocks corresponding to SHR, BN. There was go from upper left to lower right. There is one block on the HXV animals. So my point in showing this is to say there are degrees of relatedness. Even on genome-wide, they are strains, substrains, different breeding colonies of the substrain, different individuals of that breeding colony. Depending on your question, you may care about minute difference or you may not. If you choose to worry about all the possible differences or errors, then nothing can be done. So there is always a study-specific decision on which degree of variance you care about. A lot of these tiny green boxes are technical replications that indeed show you to a certain degree we have a good accurate portrait of the single nucleotide variants. Then again, technically on the side, they are out of this business side. We need to look at the color-coded one. We found discrepancies between what's recorded strain and what's the genetic relation is doing. Then we go back to say folks like, well, how helped talk with Mindy to redo bookkeeping, including some renaming of the record.
Did you redo it in Excel? Was that an Excel spreadsheet?
That was an Excel, but
Okay.
Yeah. There is explicit documentation on what it did in Excel. Yeah. Another point is many of you have seen this so-called phylogenetic tree. I put in quotation mark, which is the genome-wide view as a descriptor. Even then, on the highest level, you line up 30 or so recognizable strains, but you go to any one, you have substrains. You can go to the next level. There are tissue sources from different colleagues, and some of them are sequenced multiple times. So this tree is highly hierarchical. There are different types of errors. Some you may not care. Sometimes you want to debate what we mean by errors. So there were, again, I'll fly through a few slides about a descriptive comparison with past efforts to understand the whole genome relationship. There was a 2008 version from this community. Then there's a lot of, not Excel, but still highly descriptive notations about what's new and what's surprised. I'll switch gear to another point, that is, by the time you have genotype data, you still find regions with high densities of heterozygous sites. On the left were the findings based on RN6. Those regions are fewer and less tall in BN7, but they are still there. We call this high heads regions, but also candidate segments with potential assembly errors remaining. So this is cannot be pursued further. So the upper heat map is just on the BN animals, where the blue is the heterozygous genotype. So you see some regions, both vertically, almost all are heads, horizontally the aggregating windows. So you can treat this as a genomic track, some segmentation. You can define windows with some in-house rules. And then that allows you to also look at the non-BN samples. They sometimes have the same regions behind head, sometimes they have additional regions. Those could be genuine between strain differences, or a region that's folded in some strains but not in the BN. So now, I think it's helpful for us to speak of all these different types of variation with exactitude on what is between strain, what is a true assembly error. So now we know the non-BN samples have additional high-head regions, and then some of them may be genuine divergence from the BN, but we cannot blame it on the BN-based reference being error. So with some in-house rules, we flagged 600 regions as part of the bioinformatic blacklist shared with the community. They vary by length, but together there is 1.4% of the BN7 still out. So certainly want to be aware of this. Sometimes it comes out with list. Then in addition to that, Hao and others did a whole data set as detection. So those are changes across strains. Some of them are can be blamed on reference error. Others are genuine strain differences, genetic variation. So these two types of differences should be distinguished. In terms of regional diversity, this is no longer a wholesale genome-wide difference. If you just do IDS for the whole genome, there are 34 US strains, but this degree of similarity varies from chromosome to chromosome, and we know it's going to have no choice but to look at individual regions. The easy case is to go to all the RI panels you sequenced. So it's the top is one parent, bottom is another parent. In the middle are these RI lines. It's, of course, there are two color switches documenting for each animal the precise switch point of the contribution from the two parents. Occasion, so this is not new. Even with low density typing, you have that as part of the RGB. But we do see some animals with long tracks of heads. So these are very likely breeding errors that bred across into different lines fairly recently. So they are surprisingly head problem rather than maintained brothership community. So back to the 34 lines, I'm showing is left to right are the genotypes. I squashed away the heads and the locals. So red is homozygous ref, or blue is the opposite. You can see even for the first 1,000 sites, there are windows where there are 2 or 3 biological types, or you can say ancestral types. Even though nominally there should be 34 types. but empirically observed structure of the genome, probably just calling on this mental picture of fewer number of ancestral types further back before you're establishing these lines. But it can be 3 or 4 or 5, at which point is computable, it's not easy. So one way, well, some of you know how to code a Markov chain and then be very, very principled, but somewhat black box. What we did as a view is kind of a culture data ourselves, going to the old tool haploview, asking how many haplotypes do you see where the switch points are. So just going to one chromosome for these 34 tracks. Haploview said you have up to 16 haplotypes. It looks flickery, right? Because there are a lot of frivolous definitions of new haplotypes, but there are also, so that vertically, some needs to be scrubbed away. Horizontally, there are also frivolous switches that switch back instantly. So there are some in-house rules developed to scrub vertically so that if haplotype has 1 or 2 private variants, that could be genuine de novo variant, we don't give it the honor of a new haplotype. And then horizontally, as the next step, we kind of penalize the switching. So now the crazy quote is less crazy, but still it's a semi-principled approach to document the nameable entities in the genetic kind of evolutionary sense. So I hope the advantage is because in the past when you deal with the 8 funder mapping panel, the mathematical formula then is to do an 8-way deconvolution, but now we know region by region, the nameable entities are often fewer than 8. So few slides. And slides to show the number of haplotypes is peak at 3, 4, 5, 6, and then come down. The individual strains, you ask your top 2 haplotypes account for how much of the genome. Right. So just mysteriously appear, but let me recap some of the points. The first is it's helpful for us to talk about different types of errors. Some are playing on the reference genome that's still improvable. Others are true differences, but on different levels between strain and substrain, between breeding colonies. And then there are different difficulties because some of the questionable cause lands near indels or land resides squarely in this folded or NC from regions. Now we have the opportunity to empirically define astrotypes, and that hopefully can improve the mapping. And then there's a whole question for those who care about the host of other concepts. What do we mean by IED? There implies time in the past, before which there are different ancestry, after which you have IED sharing, and then the imputation strategies. And what are these few color painting of the 34 genomes is equivalent to constructing the graph and genome for the rats, or should we call that ancestry complementing graphs? So there are ways that we sort out. Then the roadblocks, many of you care about the functional part, so much of not so much of structure. But then your annotation may or may not be landing on this shifty part of the genome. So to be aware of that. Because like in genomics, the first 90% of analysis is easy, but the last 2% can quagmire you for many, many years. Or like rock that you're getting to the whirlpool and go down. So I'll stop there. Yeah.
Great. Then